I have some students that compose some abbreviations, and then they don't remember what is the meaning of the abbreviations.
Sometimes people just take the most random letters of sometimes sentence even, and
but
just to compose the word.
Chico likes some small animals, like Chico.
Usually
Komodo
I like
is an
to play
abbreviation of something, or just Komodo?
Abbreviation.
Abbreviation, see. Calango.
Abbreviation
also.
too.
And do you remember what calango means or Komodo means?
Calango is like
See,
Andreas, calango is like a tiny lizard in Portuguese. In some regions of Brazil, we use this word calango to refer to a couple of small lizards. And calango, I think it states for comparative analysis, and I forgot the rest. But yeah, it has a meaning.
I told you. Sure. So it's a good. What's going to be the next? Well, they called deep clusters the diamond developed for
comparative analysis with annotation-based genomic components.
You are reading.
Yeah, of course.
I will just get some water quickly. I will still hear you.
Okay. All right. Daqui a pouco a gente vai perguntar no chat de onde a galera está conectando. Você conseguiu ver alguém de outro lugar aí nos participants? Let's see if I can see someone from other places. Gabriel. By name, I think we have some students coming. Thieres, do you have a camera over there, microphone? Are you going to make many questions for Andreas?
Can you hear me?
Yes, we can hear you, Thieres.
Earphones, my girlfriend take it to the stage. I using the mic of notebook, and it's not good.
It's sounding very, very well. No problem. No problem. So, Andreas, during the pandemic, we were organizing seminars, sometimes paying the ticket, inviting people to come in. But we started with a seminar about the Trinity for assembling RNA. It was in the time of Skype. The person gave a talk using Skype. Zoom did not exist. So then when Zoom and pandemics started, so then we realized that we could invite people to talk. And so we realized that we could ask people from other places in Brazil to join the webinar. So probably today we will have people from other states also.
Nice.
So
that's very cool.
Yeah. Maybe we can ask the chat. Galera no chat, escreve aí de onde vocês estão conectando para a gente ver se só tem Belo Horizonte. I'm asking them to say in the chat where they are connecting from. Belo Horizonte is here. We are promoting the seminar today. Sometimes some people in northeast of Brazil promote the seminar, so then more people are from there. But let's see in the chat. What is the chat? I have to go full screen to see the chat. I don't see the chat anymore. Participants.
People are watching us from Natal, Belo Horizonte,
Natal.
Rio de Janeiro, Juiz de Fora.
Rio de Janeiro. More people, say where are you connecting from? Type in your cellular or your computer. Rio de Janeiro is by the sea. We don't have a contact with the ocean here in Belo Horizonte, but we have many bars and cachaça. That is, what's cachaça in English?
Cachaça is like a Brazilian, it's a spirit.
Yeah. It's a spirit, but it's only once distilled, not not double distill that would make the run. It's pretty much. That's good. Okay. Well, let's start in some 3 minutes, so then we can go on, right?
Sure.
Just to give the regular 10 minutes for them to get prepared. I think that in Germany, you have also a name for that when you are going to start sharp or not sharp.
Right, like
some people told me.
Like 15 past is a usual
15,
time to start. Yeah.
15. Ah, okay.
And I think we use the abbreviation CT, and it might come from Latin or whatever. I'm not even sure what it means, but
it's
I
very
believe.
Smart. Because then people remember 2 o'clock, let's go to the seminar. Oh my God, I have to rush. So, but then we still have some 15 minutes to get there, to have some conversation about the football. No?
Yeah, 15 past is usually easier to reach than sharp. Yeah.
Ah, okay. So just 2 minutes to go. We go look at like popcorn sometimes. We go to see the number of participants, like microwaving popcorn. So when it stops popping, we go. Usually can go. Francisco Lobo. So are you going to be the chair today?
Who, me?
Uh-huh.
Okay. So can we start now? Should we wait a couple more minutes? 10 to 10 maybe?
A couple, yeah.
So besides the time difference, there's also a season difference between us. Like there's a lot of snow outside.
There's snow outside.
A lot of snow here.
Mm-hmm.
Here in Belo Horizonte right now we have.
You are at home or by the university? Or
I'm still at the office. I just stayed in the office and then I will go home.
Okay, just waiting a final minute and we can start. Okay, Andreas?
Yeah, sure.
All right. So before Chico takes the control, Andreas, thank you for giving the talk to us. It's very important for the students to know about the software. Like it just you just produce it, so we are just going to use it. So it's going to be a pleasure to see how to use what we can do with it with Emerald, because many of people in the audience are familiar with Diamond, of course. People use here in Brazil.
Yeah, it's very big. Yeah.
Yeah. So thanks, Andreas, and Chico, you are in control.
Okay. So Andreas, thank you very much for your time, for spending a little bit of your time to present us your work. Andreas is a PhD student from the University of Helsinki, where he's located at the Department of Computer Science, Algorithm Bioinformatics. Is that right, Andreas?
Exactly, yeah.
And I understand you are part of a group who previously developed Diamond, which was a game changer in sequence alignment. And you guys use Diamond Deep Cluster. I didn't know about that. I just learned. So sorry, go ahead.
Yeah, not quite. So actually we collaborated with people from the Max Planck Institute in Tübingen, and they developed Diamonds.
And you guys use a new flavor of Diamond, which is Diamond Deep Cluster, which is used to build sequence clusters. And based on that output, you guys developed Emerald. I had a lot of fun reading your article, and I have a couple of questions for you later on. So once again, thank you very much for spending a little bit of your time to join us. And could you please try to share your screen so we can see if everything is working?
Yeah, can you see the slides? And what about the mouse?
Perfect.
Okay, great.
Everything is working. I can hear you loud and clear. I'll be here. If you need anything, just let me know.
Okay. Yeah, thank you a lot also for the invitation. So I'm presenting the work on emeralds. It's called Sensitive Inference of Alignment-Safe Intervals from Biodiverse Protein Sequences Clusters using emeralds. And this was joint work with Arthur Günther, who was a summer intern at our place back then, Fernando Diaz, Benjamin Buchfink, and Heik. Those two are from the Max Planck Institute in Tübingen. And Benjamin is also the person who developed Diamond, and also with my supervisor, Alexandru Tomescu. Okay, so let's talk a bit about optimal sequence alignments. There are some problems with them, and there are many of them. And in biology, we are interested, of course, in the correct one. But it's not always clear what is the correct one. There might be multiple optimal alignments, and just pure computationally, we don't know which one to choose for biological purposes. And also, if we have multiple sequences, then people usually decide to do multiple sequence alignments. But this is a complex problem, and people usually do some heuristics and solve many different optimal alignments between two sequences. They also require one specific choice, and then this choice is propagated through several sequences. So if there's somewhere an error, a biological error, we didn't choose the correct one, then this is being propagated throughout the next sequences. So the error even multiplies there. And also, sometimes we are not necessarily interested in an optimal alignment, but sometimes the correct solution is hidden in the suboptimal alignment space. So those are all things that we are trying to address with the tool Emerald. We are using a technique that we call safety. So we call a subsolution safe if it is contained in all of the solutions. And our solutions here are the suboptimal alignments, and subsolutions then are subalignments. We will look at what that means exactly in a bit. But here is a nice picture that illustrates it. So we want to capture those kind of subalignments that are really contained in all of them. And the intuition here is that if we assume that the biologically correct solution is hidden inside our suboptimal alignments, then the subsolutions are definitely going to be correct if they are contained in all of them, because one of them is the correct one. So let's look at the specific alignment here. Those are just two playground strings that are supposed to mimic protein. And those are all the suboptimal alignments using the Blossom 62 matrix, I believe. I forgot the number exactly. One of the Blossom matrices. And this delta here means that we are considering alignments that are at most 8 score away. And we see that here, the beginning and the end of these two strings are contained in all of our alignments. We are really looking at the pairs of the amino acids. For example, here, L and K, this pair is contained in all of the alignments. Whereas, for example, this S and this F here is not contained in already the next alignment here. And there happens to be only one optimal alignment. This seems a bit strict also. So what we also consider is to consider those sub-alignments that are contained in some alpha percents, and here we consider 75%. So this LK-LK pair here is contained in 75% of all alignments, and then we call it 75% safe. And those sub-alignments is what Emerald is calculating for us. So the goal is to find what you might call conserved regions of proteins using the safety. And those regions, we call them safety windows. And we want to scale this up to the tree of life. And this is what Emerald is supposed to do. And we want then to use Emerald to understand what do these regions mean biologically? So how are we calculating these sub-alignments? We are doing this with the dynamic programming graph to calculate alignments and optimal alignments. So every alignment of two strings corresponds to a path that goes from the top left to the bottom right in this alignment graph here. So what is hidden here is that this is a directed graph. Every edge here between the nodes goes from the top left to the bottom right, or just to the right here, or just to the bottom here. And all of these edges have a weight. These edges correspond to choosing an alignment between two characters, or doing a gap with one string or with the other string. And the Blossom 62 matrix, or any kind of alignment scoring matrix, determines here the weights for every edge. And then an optimal alignment corresponds to an optimal weighted path in this graph. And suboptimal alignments then as well to suboptimal weighted paths. So there's this one-to-one correspondence between paths and alignments. For example, here, the black path here, just aligning every amino acid, corresponds to just walking diagonally. Whereas, for example, the orange path here, we have some gaps here involved. The horizontal gaps correspond to these three gaps here. And then the vertical edges here correspond to these three gaps. So we are finding the minimal graph containing all of these delta suboptimal alignments. We calculate this one. And once we have found it, we want to find like those subpaths that are contained in an alpha percentage of all paths. So we do this by, well, this is a directed acyclic graph. So we can calculate for every node what is the number of paths going to the sink, to the bottom right node, or to the source, to the bottom left node. Those are, we calculate these number of paths. We can just do that. And then we know for every edge, what is the number of paths that we can reach the path from the source, and what is the number of paths that we can reach the sink from this edge. And we can also as well do this for subpaths. So for example, here, the safety window number 2, we just know how many paths going from the top left to the beginning of this safety window number 2, and how many paths do we have from the tail of this path to the sink. And this tells us then whether this safety window number 2 subpath is alpha percent safe, for example, 75% safe if it's contained in 75% of all these paths. And corresponding to the example before, here these safety window number 1 and 3 are 100% safe because you can already see they are contained in all of the paths that go from the top left to the bottom right. All right, this is the computational method of how to do this with pairwise alignments. And if we want to consider multiple sequences, so here we have a playground cluster that one might find with diamond deep cluster. Then we do this pairwise. So we find a representative with some methods. In our experiments, we have chosen the protein which was the highest in the phylogenetic tree, for example. And then we do this alignments with every pair of cluster representative and then all the other cluster members here. So we have 5 sequences, and then we do 4 alignments. And then we get the safety windows for all these pairs. And so for example, this M and this S here in the cluster member are covered by the safety window here, and then we mark them green on the bottom left here for the safety windows. Or similarly, this L and this K is covered by safety window number 2, and then we mark them here on the bottom left for the cluster members. So kind of mathematically, we just project them to one coordinates if you want. Okay, this is basically what Emerald is doing. So we have two parameters. We have this alpha, and we have this delta here for the suboptimality. Okay, so we kept it as simple as possible with the calculations, and we want to now give some proof of concept for the experiments. Okay, so the dataset we used is SwissProt in June 2021. Back then they were 560,000 sequences, and we clustered using, it should say Diamond Deep Clust. And we filtered out all the singleton clusters, which resulted in 16,000 clusters and 400,000 sequences. And we ran with the parameter 75%, as we just saw in the example, and also, again, data equals 8. But you can use any parameter that you want. Usually you want to choose alpha to be at least 50% for technical reasons in the algorithm. And for our experiments, we have assigned to every amino acid some structure type using the tool stride. And we define a base to be stable if it's not coil, strand, or turn. So this is just some proof of concept. It's not a perfect definition. Here's a table with all of the structure types that appeared in our data set. And the left axis here tells us what is the coverage for every structure type by safety. So how many amino acids of each structure type have we marked with green, which is part of the safety window. And the general trend here is that for the stable structure types, a higher percentage of amino acids is covered by safety compared to the unstable ones. What is missing in this figure here is how many of each structure type, how many amino acids of each structure type do we have? So in alpha helix, it's around, I believe, 40%. Bridge, free 10 helix, and pi helix don't occur so often. Especially pi helix only occurs like 0.007% rounded, something like this. So there's very little. Let me take a quick look about the other stable ones. I have it in the paper, just 1 second. Yeah, bridge appears, for example, 0.8%. Free 10 helix appears 3.2%. So they don't appear a lot, but are still captured quite well. And the total of the unstable ones appear around 55%. So with the exception of strands, we cover the stable ones better usually. And the fact that bridge and free 10 helix and pi helix do not appear so often, are very sparsely occurring in the sequences, and they're still somewhat captured by the tool, tells us that we have somewhat of a good precision with this approach as a motivation. We have more figures. We also considered the recall. So we, on the one hand, consider whether an amino acid is safe or not, and we also consider whether an amino acid is stable or not. And then this stable structure retention here, you might also know it as recall. So the stable structure retention here captures the ratio between only considering the stable amino acids, restricted on those, how many of them are safe. And then the safety coverage means considering all amino acids, how many of them are safe. And the trend here is, so here, these intervals are our sequence identities in the pairwise alignment. And the n here denotes the number of alignments And we see that maybe unsurprising, if the sequence identity is high, this means the sequences are more similar. The safety coverage is also higher. And that is because most alignments are optimal. And there is not in the alignment graph, then there will not be so many opportunities to diverge with the path. We have like most likely a single optimal alignment, and then to become suboptimal, you have to go really high with the delta. And obviously, if the safety coverage is high, the stable structure retention also grows because if every amino acid is covered, if the safety coverage is 100%, then obviously we also cover all the stable ones. But we usually see here a trend, which is that the stable structure retention is always a bit larger than the safety coverage, which means that among only the safe amino acids, there's a larger amount of stable ones than like percentage-wise than in the whole sequence, which means that we are closer to capturing stable amino acids than only looking at the full sequence. So the idea is that we want to restrict the sequence to only the safe parts and then only work with the safe parts. That's the overall intuition. Those are less amino acids in the end. What we also notice is that it even works well for smaller identities. So for 20 to 40%, we are still capturing more amino acids than if we just consider the whole sequence. And also in 40 to 70%. We have done the same experiments when restricting the sequences to those that have at most 80% safety coverage. So those yield less sequences overall. The N here is smaller, especially in the 70 to 100%, since compared to 44,000, we only have 503 now. But in the lower sequence identity ranges, even if we only look at the sequences that are covered with at most 80%, for example, here 40 to 70% sequence identity, even the median here of the stable structure retention is at 80%. So this means that we capture stable structures with safety. Of course, not perfectly. This is yet another figure. So here, let's focus on figure A first. Here we consider again all the sequences. And in the x-axis, we have the stable structure retention. We want it to be at least x. So for example, looking at 0.85, we look at those sequences, those alignments that have stable structure retention of at least 85%. And on the y-axis, we on the left figure have the safety coverage. And on the right figure, we have the proportion of sequences in the corresponding identity range. So if the stable structure retention is at least 0, then we have 100%. This is the case for all the sequences. So the proportion here is 100, but we have done anyway this cutoff at 60%. What does this figure tell us? So if we, for example, look at the dark red line here, which is all the sequences with identity range from 0 to 100, we see that 20% of the sequences have a stable structure retention of 100%, which means that in 20% of the sequences, we cover all the stable structures, but the safety coverage for them is only at 15% around, so about 5% less. Of course, this number grows with high identity sequences. And if we look, for example, at the purple graph, which is, at the purple line, which is 40 to 70% identity, then yeah, also a bit more than 20%, let's say 22% of the sequences have 100% stable structural retention, but also around the same. Only 15% are covered by safety of the amino acids. Or if we go to 80% stable structural retention or more, we see that this is the case for 75% of the sequences, but those sequences are only have a safety coverage of around 65%. So I see a message in the chat, but it's in Brazilian Portuguese. Okay, so there's something surprising also about this graph, which is that if the stable structure retention grows, the safety coverage shrinks, which is surprising because we already have the we already know that, okay, if the safety coverage grows, the stable structure retention automatically also grows. But we also have the converse. If the stable structure retention grows, which means we cover a lot of stable bases, those sequences where we cover lots of bases, we also we are precise with that. The safety coverage goes down at the same time, and I think that's quite fascinating. And in B, we have the same calculation again with the sequences restricted to at most 80% safety coverage. And here, for example, again with dark, or let's say with purple here, 40 to 70% identity, 10% of those sequences have 100%. We cover 100% of the stable bases with safety, but they have a very small safety coverage. So this is all what this figure tells us. Okay, those were our experiments. So about the tool Emerald, it's written in C++. What it does, it runs in time n squared, where n is the length of the sequences. So usually in Swiss prot around 300 maybe. And there are some advantages of it. So by how it works, we don't choose an arbitrary optimal alignment, which might or might not be correct. We only focus on those parts that are likely to be correct. If alpha is equal to 100%, then we know they are correct. If it's 75%, then we know they are likely to be correct because they are in 75% of all the alignments. So we have exact calculations, whereas in multiple sequence alignments, people use this heuristic method of considering only 2 sequences at a time. And we are doing well also in lower identity pairs, precision-wise. The source code is on GitHub. It's free software. And the tool is on Conda, or there are also some binaries for macOS Silicon and also Linux on GitHub, or you can also compile it yourself, of course. It should be easy to compile because we don't use many libraries. The only library we use is the GNU multi-precision library. So, well, relatively easy compared to many other research tools. It's easy to compile. Here is a graph about the runtime and the memory usage. So usually we didn't take more than 1,000 seconds, where every dot represents a cluster and the color represents the cluster size. So the larger the cluster, then the runtime grows linearly because we do the same operation on every pair. But if the representative sequence length here grows, then we have this n squared relation here, which also coincides with the theoretical runtime. And this is the memory usage. It can grow quite large, up to 4 gigabytes. And I believe that there's a technical reason for that. What Emerald does is it shuffles all the sequences, but then wants to print them out in the right order again. And that's why it stores the results of every alignment in memory before it outputs them all at once, because we want to use the correct order. And the reason we do the shuffling is that we can compute every alignment parallel, because those are independent computations. And we do the shuffling just so that not one thread gets all the complicated alignments and the other threads gets all the easy alignments. We want to have this fair threads calculation. Right. So the parameters you can use for alpha is at least 50%. Any delta, you can use multiple threads. And standardly, we use Blossom 62, but you can also use any custom alignment scoring. So for example, you can also consider edit distance or other Blossom matrices if you have like some specific needs for your proteins. This is how it works in the terminal. So we have a FASTA file here, some description of the protein, and then the protein itself. You can choose any representative sequence you want, but automatically it chooses the first one. And then it calculates the safety windows here. So for example, the alignment between representative sequence and cluster member 1, those are the intervals between the both strings starting at 0. It's the same example as the very first figure that I have shown you. Okay. Yeah, thank you very much. So here are some QR codes that you can scan with your phone if you want. There's a paper in genome biology if you want to read it. And yeah, I want to thank my collaborators, especially Arthur. He's amazing. He wrote a lot of the experimental pipeline, and he made my work much less stressful. And even when he was gone and I was trying to understand all of his code, it was actually understandable, which is rarely the case. So I'm very grateful to him, but of course also to all the others. Heike and Alex were also great. Especially Heike will also be here next time. Very motivated person. So they are all great collaborators. Okay, yeah, but that's it with the talk, and thank you.
So Andreas, thank you so much for the talk. I think it's me personally, when I have the opportunity to read an article like this one, where you guys are dealing with the foundations of bioinformatics, like we take for granted that the optimal alignment is the one to go with, and it doesn't need to be this way. And it was amazing. It has been a while since the last time I thought about sequence alignment. And so thank you for the opportunity to read your work. I got a couple of questions for you.
Yeah, sure.
But first, I would like to ask if any of the participants has any question before I start asking mine. Questions, everyone?
Well, I'm in the audience today, so I can ask a question. You are the chair. So,
okay.
Andreas,
you go first, Miguel.
I was wondering if we could just submit PFAM to Emerald. So if we submit all the PFAM alignments to Emerald, the output would be PFAM labeled to restrain the alignments just to the safe places, right? The safe positions.
Mm-hmm.
Is it possible to run PFAM, complete PFAM, and I would not say only PFAM, but blocks, no? Right? All the matrices that were used to deduce the Blossom 62. I was wondering if we run PFAM on Emerald and we take away all the unsafe regions, if we could generate a novel PFAM that would be dedicated more to the safe positions. Do you think that's possible to derivate a dedicated Blossom? Because
Yeah,
Blossom is not derived of chemical properties. They derived from the PFAM alignments. So I wonder if the PFAM alignments are like all safe. Probably they are not. Probably they are a portion of them are safe and a portion are not. So is it possible to run PFAM and to let us know how
so
it goes?
The thing is that we have used Blossom to find the sequence to find the safety windows, right? So I'm not sure, like
their alignment with profiles. They use Markov chains, but the alignments are there ready. I just want to run Emerald on the PFAM alignments, so it's easy experiment.
I see. Yeah, I don't see why not. I guess you might do that. So I think you would have to figure out what it means to so what exactly, like if you just want to remove the amino acids, or if you want to replace them by some characters that don't represent any amino acid or something, that maybe would have to be figured out, but I don't see why not. So this is in fact the general idea that we want to encourage people to focus only on the safety part. But
I'm wondering from your output, how, because your output for us it's like the alignments with the green labels, but for a computer it's different, right? You have independent sequences and values underneath them.
Yeah, so the output is basically just the output is basically the following. So I can show it here maybe. So
I'm wondering if you could rebuild the alignments and showing not labeled on green, but showing only the safe ones.
Yeah, I think you could do that. I'm not sure what would be the best way. Like I said, yeah, essentially the computer output here basically tells you what parts of the protein are safe. So the intervals of the protein here, and one would have to figure out what it means to align only some substrings of the protein, like fixed together, patched together, what it would mean, this alignment. But
usually the unsafe we substitute by dashes.
Exactly, yeah, you can substitute the unsafe ones by some random character that doesn't represent anything, and then do the alignments. I think this.
No, I mean after the interpretation, I want just to substitute the ones that I don't care by dashes.
Yeah, exactly. Yeah.
And then use this alignment in other tools, like to do some phylogeny, etc.
Exactly. Yeah. That's in fact what we are doing with in another project right now as well, we have substituted the unsafe ones with some characters, and then we look at we compare the results between the full sequence and only the safe ones. Yeah, this totally works.
Could be fun to run a phylogenetic tree analysis based only on the safe ones to see how it goes.
Yeah.
And my other question, Chico, how do you generate, is Emerald generating the several options of alignments? Is this inside Emerald? You don't use like muscle or cluster or whatever to run the alignments. Emerald does the alignment.
Yeah, we just wrote it ourselves, yeah.
It's basically a Needleman-Wunsch algorithm.
Yeah, exactly. Yeah. It's so it's doing so Blossom. It's using a fine linear gaps. So instead of this graph here, what we actually have is 3 nodes inside every node here, and then edges between them. And exactly, it's basically this Needleman-Wunsch finding optimal paths. You
need to have table. Like 2 matrices for that, right? The matrices with the vectors pointing where you should go, and the matrix containing the alignment score. And based on that, you can find your path.
Yeah.
Okay, so before I start asking a couple of questions, I think Gabriel has one question for you, Andreas. Gabriel asked if you think Emerald could be used to differentiate between very similar conserved domains, because since it's based on stable structures, Gabriel wonders if you can find conserved domains using Emerald.
So this is also something we are interested in. So we are working on right now whether Emeralds can tell us something about different domains of a protein or not. So this is actually ongoing work. But the results so far look promising that it can actually give us information about the domains, whether there's only a single domain or multiple ones, or if there's multiple 3-dimensional structures or a single one. This is something we are using Emerald for.
I think the benchmark strategy you guys used is very clever, because you're looking at secondary structures first. But the next step I would think about is, okay, if you can find secondary structures, they are contained preferentially within the safe regions. What about domains? That's
exactly, yeah. This is something we are working on at the moment, using Emerald and Diamond Deep Trust as well.
Cool. So I got a question about this figure. When you say, I understand you need to provide 3 parameters for Emerald. You need to provide alpha, delta, and the matrix you are hoping to use to align your sequences. Is that right?
Yes.
Okay. This is kind of a philosophical question. So you gave me here an optimal alignment, but this optimal alignment is based on the matrix I'm using, right?
Yeah.
If you use Blossom 62, you're expected to have 1 optimal alignment. If you use Blossom 40, which is for more divergent sequences, you may have other alignments, right?
Yes.
Okay, because, I was reading your article and I was always wondering, maybe we are too aggressive when we choose the values to open a gap or to extend a gap. You know what I mean? Because in this picture here, you can see that the difference between why the optimal alignment here doesn't find the safety window number 2. Because I was wondering, I don't know if I'm right or not, but I was wondering that maybe this is because the cost to open the gap is too high, and so the Needleman-Wunsch algorithm will choose this one as the optimal alignment. So maybe if you play around with the cost to open and extend gaps, you may get other optimal alignments where gaps are not as costly as they are right now in the default parameter. Have you guys played a little bit with other scoring matrices or playing with the cost to open or extend the gap?
We have not really. Yeah, we have not really. In Blossom 62, it turned out that I don't remember anymore how many percent of the safety windows were actually contained in the optimal alignment or not. I think I figured, I think I calculated at some point, but I don't remember. But we have not played around with this, but I think it makes sense to experiment with this and see what gives the better results. We have rather chosen different parameters with alpha and delta, but just fix it with Blossom 62.
Yeah, because I mean, if you are, go ahead, please.
Yeah, I think the majority of safety windows are actually contained in optimal alignments. But as you said, with other matrices, it might be that more of them might be in suboptimal alignments. But I think in our experiments with Blossom 62, it turned out that the majority of safety windows were anyways in optimal alignments. And of course, this example here is handcrafted so that the graph looks nice. And
very easy to follow. I mean, I understood how your algorithm works, only taking a look at this picture. I think it's very self-explanatory. But you got what I was wondering, right? In the optimal alignment, you don't have any gap because the cost to open a gap or extend one this is very costly. But if you change that a little bit, so sorry, go ahead.
Yeah, this is also why we consider this delta value here, because as you say, the suboptimal alignments might matter. So one way to mitigate this is through adjusting the gaps. But we just chose to solve this issue by increasing this delta value. Which here, I mean, in the graph itself, we don't differentiate anymore between optimal and suboptimal. Those are all just our alignments, right? So this is kind of our way of how to deal with this issue, I guess.
Cool. Okay, we got another question. So Eduardo Horta is asking, first he's saying, great talk, Andreas. I agree.
Thank you.
Very, very nice talk. And Eduardo was wondering if one could use Emerald as a confirmation or failsafe in making novel protein functional annotation. How do you think? So he's asking a couple of questions. I'll ask the first one. So he thinks if you can use Emerald to confirm or to make novel protein functional annotation.
Yeah, I mean, this is basically one of the dreams. I mean, so far we have only shown some proof of concepts, and this is something I would be interested to figure out with potential further projects, or also see other people try to do. But I mean, looking at the experiments, we want to motivate that this is something we believe is possible.
Cool. And he also asks, what do you think are the implications of using Emerald in this context, and how applicable would be using it in a functional annotation pipeline?
Yeah, so okay, with the for example, figuring out understanding the domains better of a protein is something that leads to good results. So I believe that using Emerald probably is going to be worth it to annotate proteins. Something we are also dreaming about is understanding, for example, AlphaFolds a bit better, because it's hidden behind this machine learning abstraction, and it's difficult to understand what is actually happening. And yeah, we are doing this right now, and we are seeing good results. So I believe that it's a good idea. I'm not sure how exactly to interpret the last question. But one application is, like Miguel suggested, one way to apply Emerald is to focus on only the safe amino acids. And something we have not done in this toy experiments, in these proof of concept experiments, is we have only looked at whether an amino acid is safe or not at the intervals themselves. So this is something that you could do in pipelines. And so, for example, if you wanted to replace multiple sequence alignments, which, if you want to do it optimally, does heavy computations on the whole sequence, what you can do now is you can first do the pairwise alignments and then do the heavy computations on the restricted part of the sequences that we actually care about.
Cool. Yeah.
I hope I answered the question well enough.
Eduardo says that, yeah.
You did not think on doing hierarchically, right? So like the two most similar ones, then you add the other one.
Actually, no, we have not thought about this.
But it's pairwise.
It's anyway pairwise, so we do all of them. The order of when we align.
They are anchored by the reference.
By the cluster representative, which
exactly, yeah.
So that's another question, Andreas. So
yeah.
I understand that Diamond Deep Cluster uses the centroid as cluster representative, but you said during your talk, and I was thinking that Emerald did the same, but you said during your talk that you use like a real sequence for that. I mean,
right,
for the cluster representative.
Yeah. So the cluster consists of all the sequences here. So the cluster consists of these 5 sequences, and then we choose one of them. So if the cluster has n sequences, we do n-1 alignments. Yeah, exactly. That's another idea that we actually haven't considered. Using some centroid, some average, or I'm not sure. Yeah, that's a good idea also. Yeah.
Is it too difficult, too consuming to actually to use all sequences as representative, because you do pairwise, or you go pairwise with the reference, right? So if you wanted to do all to all, every sequence would be the representative, but at its turn. And then you should have to assemble the results to have a full measurement. Using all of them as representatives,
right?
Would that take a long time to compute, maybe?
Well, I guess
if you could do in parallel, then
yeah. I mean, it depends on the cluster size. If this would then grow quadratically with the cluster size, so it would take. Yeah, I'm not sure how big the clusters can actually be, but I think they can grow quite large.
Yeah. Technically, it can be done, but maybe it takes a long time, and maybe you could provide a heuristic to reduce this time. But I wonder if you would get another type of final result. If you
that's true.
Everyone as a representative and assemble the result for the cluster.
Exactly. So one would then have to figure out how to exactly assemble them.
The distance
to one big results. Then we have quadratically many of them, and we somehow need to combine them. But
you have
there are many.
To apply Emerald to Emerald. You do the same thing. You analyze if under this experiment, this one, the second sequence one being the representative, what's going on? What's going on if you do with the second two as representative, then you put all together and then you run Emerald on this. You are asking,
I think there are
what is safe.
Yeah, I think there are many different approaches to this.
It's interesting to anchor like a human and then refer everything to human, but it would be nice to have a measurement of the cluster. The median sequence could be one, but I would be more happy with everyone being the reference and then assembling the results at the end, maybe.
Yeah, I totally understand that. Yeah, it's maybe one of the biggest downsides is that what does this representative even mean exactly? So yeah, but this is exactly something that you can do using Emerald. So we have decided to keep it simple. So something like this can be done using Emerald, and those are just multiple Emerald calls, right? So yeah, you can choose any representative, and this is definitely something you can do. One just has to figure out what the best way to assemble these different results, to patch them together somehow into one.
I think that many alignment tools like Cluster, when you introduce the bootstrap, actually you are considering many individual alignments. Like if you could recover all the individual alignments of Cluster before doing the bootstrap, you could also feed Emerald with an alignment produced by another tool.
Can you repeat the question?
Like if you provide Cluster as the alignment tool, and if you recover all the alignments that Cluster generates before you use the bootstrap to consolidate all of them, you could use the alignments produced by Cluster, the multiple alignments and different alignments, to use as an input of Emerald. So what kind of format do you need for the input? Not your input is just the cluster, but then if we do an echo, like if we print out the intermediary result, you probably have all the possible alignments in the code. Right, at some step you have all the possible alignments over there. Can we trade this? Can we substitute this using Cluster as the alignment tool, and then we put all the alignments in this point and then proceed with Emerald? What would be the format for us to jump to the point where the alignments are produced by another tool, not by the way you do?
So that's a very good idea, in fact. This is not something I thought of. Of course, you can manipulate the C++ codes yourself, but it's not possible by just giving parameters to the tool. So it's not implemented yet, but that's a very good idea.
It's just a description of the format that you use already, maybe. So then we could jump to the middle of Emerald and proceed.
It's a very good idea, yes.
Yeah, you could try Cluster. I don't know how to recover the Cluster alignments before the bootstrap. I use Meg as a tool, and then I choose Cluster for the method or muscle, and then bootstrap 1000. So but I never see where the multiple, the 1000 tries are. I just see the final alignment after bootstrap. So Yeah. Okay,
Okay.
Chico.
Any more questions? Anyone? No? Okay, Andreas, I got one last question, if you don't mind. Okay, so I understood that you need to provide a FASTA file containing all sequences for a cluster. And in the end, I get the sequences and the coordinates corresponding to the safety windows, right?
The intervals of the string.
Yeah,
yes.
the intervals of the string, yeah. So because I work with positive selection, like I need to use protein alignments to get codon alignments to search for, to compute the how many synonymous changes and how many non-synonymous changes. Because I mean, this kind of strategy that Emerald provides us, in the end, what we are trying to find when you align sequences is to find homologous regions, right? Homologous columns within your alignment. So if I choose a given value for alpha and delta, those coordinates I get in the end, I can just use them and assume they are homologous regions to use them for any downstream analysis, for instance, for phylogenetic analysis or for positive selection.
I'm not sure what homologous means in this context.
Homologous means that that column was present in the last common ancestor of those genes.
I think that the sequence, the entire sequence will be homologous, but you are looking to the more important, more conservative, because
they don't need to be homologous, because you may have recombination, horizontal gene transfer, exon shuffling. There are many genomic changes that may join sequences that they are not homologous.
Okay, but maybe the deep cluster tries to recover groups of homologous proteins, the input that they are using there. But related to your work, Chico, Andreas, probably the conservation of DNA on those safe regions are more important than the unsafe ones. You know, GNDS, Chico, could be that you have a good difference. The safe ones should report the conservative changes more than the other ones.
Yeah, I was just wondering because
filter out some noise.
Yeah, I definitely will give Emerald a try. I mean, I have some groups of some sequence clusters that I know they are homologous, and I'll try to use like a standard sequence alignment and Emerald to see what happens. Yeah, I'll give it a try, definitely. I love this strategy. I love the software.
Yeah,
I think it's a very
that's great.
It's very creative. Congratulations.
Yeah,
thank you.
I think I would have to play with your output, because I understand Emerald as a filter. Then the way you represent the output, maybe I would like to get back to the FASTA files, because you know they are individual FASTA files. If we replace the unsafe ones with dashes, I can use a tree generator to make a tree. Just playing with this output, you can have an input to a tree generator for phylogeny, based only on the safe positions.
Yeah, that's I'm glad to hear that that you like this.
Usually we use some the FASTA format for alignment is the sequences on FASTA format like you are presenting, but then we mask the ones that are unsafe, and then Actually, this is not an alignment, right? And the alignment has to have all the. I don't know how to do, but I have the idea how to play with your output to try to do a hierarchical cluster that would be a tree. Right?
Yeah, and please feel free if you want to use the tool and you have any questions or problems, please reach out to me.
So great,
I'm more than happy to
you.
help.
Yeah, I'll drop you an email in a couple of weeks, definitely.
Yeah, that was a wonderful thing because we have the author of the program, we saw the program, and if we have questions or further developments, we can ask you. So people, you can find easily the contact in the internet of Andreas, right, Andreas? I did not place, I don't know if we can place in the chat here, but it will be easy.
We have the QR codes.
Yeah. Okay.
Yeah,
Andreas.
exactly.
So thanks a lot, and
thank you. Obrigado.
You are invited in March next year to come together to see the presentation of Deep Class.
Yeah, sure. Yeah, that would be lovely. Thank you, everyone.
Thanks a lot.
Thanks, Andreas. Bye-bye.
Have a nice weekend. Bye-bye.
Audience.
Wish you the same.
Okay. Next week, no seminar because it's a holiday here or something in Brazil. É um feriado, né, Tiago? Sexta-feira que vem. E na outra, a gente tem ainda uma sessão de um doutorando e um mestrando. Aí vejo vocês daqui a 15